We are very happy to welcome Bevilacqua, Professor of Chemistry at Penn State. His research group is centered on understanding the function of systems involving ribonucleoacids RNA at the molecular level. RNA is a fascinating molecule because it has both genetic and functional capabilities. This has led to the notion that RNA was particularly important in emergence of life on Earth, the RNA world hypothesis. He is attacking the RNA world hypothesis on all fronts with a diverse approach to research utilizing everything from molecular biology to computational techniques. He also leads the RNA Club at Penn State. In addition to these achievements, he is known to run a very fun and a cooperative lab. Please join me in welcoming Dr. Bevilacqua.
Okay, thanks very much. So it's a pleasure to be here, and I want to tell you a little bit about RNA and how we look at it and how we think about RNA, both from a molecular view and from a functional view. And I'm a chemist. I'm in the chemistry department, and so the view that I'm going to give you of this biological molecule is going to be from a very sort of molecular point of view. Okay. And so, but I want to sort of begin with motivating why we're looking into it, and that is really because it's a functionally interesting molecule. And what I mean by that, it has some very interesting biology that RNA can do. And so this begins with sort of one of the center ideas of biology, which is the central dogma. And this shows how information flows in biology. And you start with DNA over here on the left, and the information, genetic information, of course, is stored in DNA, and that's transcribed into RNA, which is then translated into proteins. And the view is that proteins have all the functions. They're the catalysts. They catalyze reactions in the body. They speed up their rates, and they lead to specificity. And then DNA over here is important because it stores the genetic information. And then RNA sort of sadly sits in the center here as the messenger between these two important molecules without really an important function itself. But the newer view of the central dogma is that in the center it has central importance, and that RNA itself has many functions. Non-coding RNA, for example, can fold into these very complex shapes. And as a chemist, we believe that this functional complexity comes from some kind of molecular complexity, in which RNA can fold back on itself and create clefts that can bind small molecules like ATP to go on and have various functions. Not only is RNA a functional molecule, but it's also a genetic molecule. It can store genetic information. Many viruses and retroviruses, such as HIV, have their genetic information in the form of RNA, which can then be turned back into DNA by reversing this first step here in a process called reverse transcription. This is not possible from proteins. We can't reverse translate a protein into an RNA. And so in recent years, many functional RNAs have been discovered and enumerated. And I list some of them here. And this is probably about 8 or 10, but over 20 or 30, if you check Wikipedia, are now listed as different classes of functional RNAs. And so RNA has many important roles in biology. So how does this come about? Well, the first thing I want to tell you is that actually RNA is quite simple from a molecular point of view. Okay? So let's start out first by looking at the other side of the coin, which is at proteins. And you can see here are all the side chains that make up a protein. There's 20 different so-called amino acids, and I've drawn boxes to sort of classify them into different groups. There are aliphatic, kind of greasy side chains up here. There are ones that have electrostatic charge. They can be positively charged or negatively charged. There are kind of planar flat ones, aromatic amino acids. And then there are polar amino acids, as well as some that are cyclic, okay? So if you're a chemist and you want to build a really good molecule to do some kind of function, this is a great toolbox to have. And not only that, but some of the side chains are special in that they can have, they can do special chemical reactions. So for example, this amino acid is called histidine, and it can transfer protons, and that's really important for chemistry. And this one is positively charged, as you can see, and that's important for certain reactions as well. Okay, so that's proteins. Okay, so that looks really good for functional complexity. This is what RNA looks like, and you can see it's much simpler. There's only 4 different side chains to build the functional molecule from. Not only that, but they look really similar to each other, don't they? Okay, there are these nitrogenous heterocycles, and 2 of them are kind of the same size and a little bit bigger, and 2 of them are a little bit smaller. Okay, so the question really is, what kind of functional complexity can one build out of this very simple molecular basis? Okay, so it turns out that it's folding that really drives the higher order complexity and molecular complexity that is, of course, needed to gain the functional complexity. So let me show you what I mean by this word folding. So, so you're all familiar, I'm sure, with DNA. And when you think of DNA, this is probably what comes to mind, which is a right-handed double helix. And if you ask whether or not RNA can fold like that, the answer is yes. In fact, double-stranded RNA is quite important, and we're realizing this over the last decade or so. But if you ask whether or not RNA can fold like this, okay, which looks much more complex, I'm sure, and you can see that it's kind of globular and it has a cleft where ligands can bind and maybe chemistry can happen, and we ask, can RNA do this? And the answer to that is also yes, and that's where some of the most interesting function comes from. So how can such a simple molecule, which has such simple building blocks, fold in this very complex way, okay? Well, it turns out that one of the key things that differentiates RNA from DNA, there are some differences, atomic level differences. It turns out there's an extra oxygen in RNA, and there's a methyl group difference. But what's most important is the fact that RNA is only composed of 1 strand. It's not double-stranded in general, okay? And because it has only 1 strand, it folds back on itself, and it contains both double-stranded stretches like here and single-stranded stretches like up there, okay? And so you can come up with all sorts of various combinations or building blocks. We call this secondary structure, okay, that come together to make the tertiary structure. So it's this structural complexity that leads to functional complexity. And these are just a few examples of the type of complexity that you can build out of this simple molecule, okay? So I said it's single-stranded, but it's not single-stranded in a simple way, okay? So you can follow the backbone here. It kind of comes up, it goes over, and goes around, and over, and back over, and around, and down, okay? But anyhow, when you look at the structure of the RNA, and this is from a crystal structure, you see that it is quite globular, and it has an inside and it has an outside. It's very protein-like, okay? And kind of one of the most important features, I don't want to go through the details too much, but that hold this RNA together, despite the fact that it's very negatively charged, every monomer in this polymer is negatively charged, is that there are metal ions that come in, and they're kind of the glue that allow these phosphates to come together and not repel each other and blow the molecule apart, okay? So there's tertiary structures that are quite complex. In fact, here's one of the most complex tertiary structures that we know of, and this is the crystal structure of the ribosome. This is the enzyme that actually builds the proteins from the RNA, from the messenger RNA. And this structure was solved about 10 years ago, and it led to the Nobel Prize in chemistry from this group, Tom Steitz's group, and several other groups. And this is just one view. It's 2.5 million molecular weight, so it's enormous. It was quite a feat just to solve this crystal structure. But from my point of view, what's most striking about this is that when you look at the active site, and this is where chemistry happens, it turns out that it's all red and white near the active site. And the red and white is RNA. So going in, the expectations were that proteins would do the chemistry and RNA would hold it all together, when in fact it's exactly the other way around. The proteins hold RNA together, and the RNA does the chemistry, okay? So it's inside out from what we expected. And in fact, the authors note that in their abstract. They say proteins are abundant everywhere on its surface except in the active site where the peptide bond formation occurs. Okay, so I'm going to give you just a few examples of functional complexity from molecular simplicity in RNA that comes out of my lab's research. And this is one particular catalytic RNA. So RNA can be an enzyme. Up until about 1980, it was thought that all enzymes were proteins, but it turned out Tom Cech and Sid Altman discovered that RNA could be an enzyme. They won a Nobel Prize in chemistry for this discovery. And this is one particular enzyme that we study. It also is single strand, and you can see the fold is quite complex. And here is the crystal structure of that. And there's one particular base that I just want to show you, the cytosine, that has all these intricate interactions and hydrogen bonds with phosphates and so on. And it turns out that when we looked at the function of this molecule, we got a big surprise, and that is that it has a pKa that's very histidine-like. It's right at biological pH, and that was very surprising. How could that be? It's almost histidine-like. So now you've got these really simple building blocks doing things they shouldn't do. They're acting like histidine. They're acting like a protein. So what's the answer? So we thought, well, maybe it has to do with the three-dimensional fold. The higher order folding somehow induces that side chain to act like a histidine. Okay, so we worked with our collaborators at Purdue and Barb Golden's lab to crystallize the RNA and then study its properties so that we could get a molecular level view of what was going on. And those are just some of the crystals. And then when we got these crystals, one of the things we did was Raman spectroscopy, which is a way of using light to understand the transitions and the properties of the RNA. And we do this in collaboration too with a group at Case Western, Paul Carey's group. And we can take the RNA and put it in a microscope in a crystal and shine laser light into it and then look at the scattering of that light and study how that changes, say, as a function of pH or something like that. And by doing that, we were actually able to remeasure this ability of the RNA to bind a proton. And in fact, we were able to find that the ribozyme, the catalytic RNA, also by direct observation has this pKa almost at biological pH, okay, in these crystals. And so we wanted to then ask how this could be at the molecular level. And so we solved the crystal structure with our collaborators. And one of the things that we found was that inside the active site, and this is that cytosine that has this biological ability to bind protons or pKa, that there's a very, very negative atmosphere in the neighborhood of that side chain. And what that does is it makes it very favorable for this to be positively charged. In fact, it has to be positively charged to allow all that negative charge to come together. So you have this higher order folding, coaxing a simple, molecularly simple molecule to have protein-like properties. Okay, and that shows just some of the interactions from those phosphates that create that negative environment. So what does this mean for the so-called RNA world? Okay, well, this is a little Venn diagram about how we think about the RNA world. And the RNA world hypothesis is that RNA was the biopolymer in the first cell. So for the emergence of life, how did life begin? You've got, if you want to think about proteins, they have lots of very sophisticated and complex functions, but they can't store decodable genetic information. And DNA doesn't have any function. So we've got RNA that kind of both has the function and it has the ability to have decodable genetic information. So it's a very versatile molecule. And when we study RNA world and we think about the RNA world hypothesis, what we can do is look at, from a chemist's point of view, what sorts of interactions are possible. And then within what's possible is what might have been in the RNA world, and then what we currently have in a world that's made up of RNA and proteins, which we can study, like our ribozymes, our catalytic RNAs. And we can ask whether there's might be any overlap in how the transition happened between these two worlds. And this is just a kind of one last kind of molecular view that leads to some support for this RNA world. And that is when we look at a lot of the important cofactors in modern metabolism, they're actually made up of RNA side chains, okay? So these are 4 different examples, ATP, and the side chain, adenosine diphosphate, is colored in blue and red and yellow, and you can sort of see how that remains in these very important side chains, suggesting that this may be some kind of remnant from an RNA world. So kind of leave you with a different way of thinking about RNA's role in biology in the RNA world. And these are some drawings of Picasso of his bowls. And this is kind of Picasso's evolution in drawing the bowls from kind of a very sort of realistic and complex way to a more abstract way to a very simple way, but you can still recognize that as a bowl, and you can immediately recognize it as Picasso. And then thinking about macromolecular evolution, you really think about it from a very simple point of view, where you kind of have RNA over here, which isn't quite as sophisticated in the functions of modern day proteins, but you can recognize those functions, and you can see the molecular framework that leads to this more complex and modern view of biology in terms of proteins. And then I just want to have 2 slides right at the end. The work that we do at Penn State is highly collaborative and relies on collaborations with a number of outstanding groups, including a group at Purdue, Bart Golden's group, working on crystallizing the RNA and their structures. We've done a lot of calculations, including the ones where I showed you the pictures where the electrostatic environment with Sharon Thomas-Schiffer, who's at University of Illinois, and the Raman spectroscopy on the crystals was done with Paul Carey at Case Western. And then this is just a picture of my research group, which is comprised of all the way from a number of undergraduates right on to graduate students and postdocs. So thank you very much for the invitation.